New approaches for vaccine delivery. Traditional vaccines are usually administered through injections, intramuscular or subcutaneous, but new approaches are being developed to make vaccination safer, easier, more effective, and needle-free. 1. Mucosal vaccine delivery. Oral/nasal/sublingual routes. Principle. Delivers vaccines through mucosal surfaces, example oral cavity, nasal passages, intestines, to stimulate local mucosal immunity, IgA, and systemic immunity, IgG. Examples. Oral polio vaccine, OPV. Nasal influenza vaccine, FluMist. Advantages. Needle-free and painless. Induces both mucosal and systemic immunity. Easier to administer and suitable for mass immunization. Reduced risk of needle-borne infections. Limitations. Antigen degradation in mucosal environments, enzymes, pH. Difficulty achieving strong immune response for all antigens. Requires special formulations or adjuvants. 2. Transdermal, skin patch, vaccine delivery. Principle. Delivers vaccine antigens through the skin using microneedles or patches that penetrate the outer skin layer without reaching nerves. Examples. Influenza microneedle patch. Measles microneedle patch, in trials. Advantages. Painless and self-administered. Stable at room temperature, no cold chain. Targets abundant immune cells, Langerhans and dendritic cells, in skin. Reduces medical waste, no syringes. Limitations. Limited dose volume, only small amounts can be delivered. Still in development for many vaccines. Cost of patch manufacturing can be high initially. 3. Lipid nanoparticle, LNP, delivery systems. Principle. Used mainly for mRNA and DNA vaccines, lipid nanoparticles protect nucleic acid cargo and facilitate entry into host cells. Examples. Pfizer, Biotech and Moderna COVID-19 mRNA vaccines. Advantages. Highly efficient delivery of genetic material. Enhances stability and uptake by cells. Allows rapid vaccine design and manufacturing. Limitations. Requires cold storage. Potential inflammatory reactions from lipids. High cost and technical expertise needed. 4. Viral vector-based delivery systems. Principle. Uses a modified, harmless virus as a carrier to deliver antigenic genes into host cells to elicit immune response. Examples. Oxford, AstraZeneca COVID-19 vaccine, adenoviral vector. Johnson & Johnson COVID-19 vaccine. Advantages. Strong cellular and humoral immune responses. Long-lasting immunity. Can be used for multiple antigens. Limitations. Pre-existing immunity to vector can reduce efficacy. Complex production and storage. May trigger unwanted immune responses to vector. 5. DNA vaccines with electroporation. Principle. DNA plasmid encoding antigen is delivered into cells, often aided by electroporation, electric pulses that enhance DNA uptake. Examples. ZyCoV-D, COVID-19 vaccine developed in India. Advantages. Stable and easy to produce. Induces both humoral and cellular responses. No need for live organisms. Limitations. Lower immunogenicity compared to live vaccines. Requires electroporation device. Long-term safety data still limited. 6. Nanoparticle-based delivery systems. Principle. Uses polymeric, liposomal, or protein nanoparticles as carriers for antigens or nucleic acids. Examples: virus-like particle, VLP, vaccines, example HPV vaccine. Liposome-based malaria vaccines. Advantages: protects antigen from degradation. Targeted delivery to immune cells. Can be designed for controlled release. Limitations: complex formulation process. Potential toxicity of nanoparticles. Costly large-scale manufacturing. 7 edible vaccines. Principle: antigenic genes are expressed in edible plants, like potato, banana, tomato. Immunity develops after eating the modified plant. Examples: experimental hepatitis B and Norwalk virus vaccines in potatoes. Advantages: very low cost and easy distribution. No need for needles or cold storage. Suitable for developing countries. Limitations: difficult dose standardization. Low antigen expression in plants. Public acceptance and regulatory concerns. 8 inhalable aerosol vaccines. Principle: delivered through inhalation, vaccine particles reach the respiratory tract and induce mucosal immunity. Examples: aerosolized measles and COVID-19 vaccines, under trials. Advantages: painless, needle-free. Targets respiratory pathogens effectively. Easy to administer for mass vaccination. Limitations: requires specific particle size for delivery. Stability and storage challenges. Limited number of approved inhalable vaccines. 9 biodegradable polymer-based delivery systems. Principle: encapsulates antigens in biodegradable polymers, like PLGA, that allow controlled and sustained release over time. Examples: experimental single-shot vaccines for tetanus and malaria. Advantages: prolonged antigen release, booster-free vaccines. Reduces need for multiple doses. Protects antigens from degradation. Limitations: complex formulation. Difficult to control release kinetics precisely. Potential for polymer-induced inflammation. 10 needle-free jet injectors. Principle: uses high-pressure stream of liquid vaccine to penetrate the skin without needles. Examples: jet injector-based influenza and hepatitis vaccines. Advantages: painless and avoids needle injuries. Reduces biohazard waste. Suitable for mass vaccination. Limitations: high initial cost of devices. Requires maintenance and proper calibration. Not suitable for all formulations. Summary. Approach. Key feature. Main advantage. Main limitation. Mucosal. Oral/nasal delivery. Mucosal plus systemic immunity. Antigen instability. Transdermal. Microneedle patch. Painless, self-use. Limited dose volume. LNP. mRNA delivery. Efficient, rapid. Cold chain required. Viral vector. Gene delivery via virus. Strong immunity. Pre-existing immunity. DNA plus electroporation. Genetic vaccine. Stable, dual immunity. Needs device. Nanoparticle. Smart carrier. Targeted delivery. Complex production. Edible. Plant-based. Cheap, easy. Dose inconsistency. Inhalable. Aerosol route. Needle-free. Stability issues. Polymer-based. Slow release. Fewer doses. Complex control. Jet injector. Needle-free. Safe, fast. Device cost. Thanks for watching.